Thank you, Professor Lo Scalzo. We'll see you later. Just the time for us to talk about the second conference. Professeur Casimir, la deuxième conférence va nous projeter dans l'univers de l'intelligence artificielle et du machine learning. Sur le plan thérapeutique, les données traitées sont source de beaucoup d'espoir et aussi de nouvelles applications cliniques. L'orateur que vous allez nous présenter est belge. Il s'agit du professeur Ivan Sijs.
Yes, our next speaker is Ivan Sijs, co-laureate of AstraZeneca Prize in 2020, data mining and modeling for biomedicine from the University of Ghent. Ivan Sijs obtained his PhD in computer science from Ghent University in 2004. After stays abroad at the University of the Basque Country and the University Claude Bernard at Lyon, he returned to Belgium, where he founded the Data Mining and Modeling for Biomedicine Group at University of Ghent. Currently, he holds a professor position at the Department of Applied Mathematics, Computer Science and Statistics at Ghent University, and is a principal investigator in the VIB Center for Inflammation Research. The Sijs Lab is widely recognized for its expertise in machine learning methods for single-cell biology, mainly applied in the field of immunology. Research in the lab spans the whole spectrum of methods and technology development up until application in the clinic. The title of his lecture is Machine Learning, Clinical Application of Artificial Intelligence, Single-Cell Bioinformatics. Professor Sijs, it's yours.
Thank you so much, Professor Casimir, for the nice introduction. I changed a little bit the title to make it more comprehensive. So in my lab, we are indeed using methods from the broad field of artificial intelligence to get more insights in data that we mainly obtain using single-cell technologies, and we're using that for a wide variety of applications, mainly in immunology. And we're also trying to go one step further and actually translate then all these AI methods to the clinic so that they also become useful for the patients in the end. So let me just start by framing a little bit the broad research domain of artificial intelligence. So in artificial intelligence, we try to build systems that do things that we as humans would think require some form of intelligence. In order to do that, we mostly use engineering and mathematical approaches to build these kind of systems instead of really trying to simulate how exactly the brain works. And the key question within the field of artificial intelligence is actually how to find good representations of knowledge and of data that actually allow us to reason with these representations and do inference, right? So infer new knowledge from existing data and existing knowledge. And so the talk of Professor Lo Scalzo was a very nice illustration of one type of representation, namely networks and graphs, and the powerful things you can do with such kind of representations. So let me just give a quick overview of the different subdomains in artificial intelligence. I'm not going to go into detail into all of these subdomains. I'm pretty sure you are familiar with many of them, like, for example, search and planning, robotics, computer vision, and so on. So these are all different subdomains. Of course, many of them hold great potential for applications in the health and clinical domains. But what I'm going to focus on today is one subdomain of artificial intelligence that we actually call machine learning. And machine learning is about models that we construct from data. So if you think about the classical way that a computer is programmed, you typically write lines of code where you explicitly say to a computer, do this, then next do that, and so on to solve a certain problem. Now in machine learning, we're going to apply another type of programming of a computer, not saying explicitly what the computer needs to do, but rather what we want the computer to learn by showing examples to the computer. So for example, if you're interested in building a system that is able to recognize faces, you're going to show faces to the computer and tell them this is a face of this person, this is a face of that person, and so on. And by doing that many times, you would hope that the computer would actually learn from these examples a general representation of a face, for example. Of course, we can do very similar things for diseases, right? So we can represent to the computer a lot of different types of data, for example, that we collect from healthy patients and patients with a certain disease. And then the computer has to learn from these examples by using some kind of mathematical models that we typically use. We're not going to say explicitly to the computer what is the difference between the healthy and the diseased patients. The computer has to actually learn these differences just by us giving examples. And there are many types of machine learning methods that we can actually use. You can see them at the bottom of the slide here. So for example, one type of methods is about really learning structures in the data. So for example, if we have a set of patients having a certain disease, maybe there will be subtypes of these disease patients having maybe different mechanisms or different subtypes of the disease. And then you would want to learn this substructure that is present in your data. So those are typically called unsupervised models. Typical application are clustering techniques where we try to find subgroups, for example, of patients that are more related to each other within the subgroup than between the different subgroups. A second type of models are predictive models. So in these models, you try, for example, to learn a diagnostic model that is able to predict for a new patient if the patient has a certain disease. And this model is actually trained typically on historical data that you have been collecting in the past. A lot of these type of models are using what is now called deep learning models. So these are typically quite complex neural network type of models that have been shown to give very good performance, especially in imaging related problems. And then there's a last category of models that is about learning policies. So you can actually compare that with a child, for example, that has to learn to bike. As a parent, you're not going to say explicitly move your feet 5 centimeters up and down and so on. No, the child has to learn by itself, by falling and doing how to bike. And similar types of methods for learning such kind of control policies can also be done using machine learning methods. And they are typically used a lot in the robotics field, for example. So in my lab, we are using these machine learning methods to mainly analyze different types of single cell data. And the single cell field is moving very, very fast. So we are able now to profile in very detail different types of tissues of a patient. And of course, by doing that, we get unprecedented amounts of information that give us indications of really, the molecular workings of cells and tissues. And from that, of course, we hope to extract some mechanistic insights about how all these systems are working and how they're differing in healthy state and disease state. So of course, if you're looking for biomarkers, for example, for certain diseases, you're looking, for example, to easily available fluids, like, for example, blood. My apologies. And you can try to extract some features from that, for example, could be used in a diagnostic model. So the types of single cell data that we are working with, well, historically, we started with cytometry-based approaches because I'm mainly an immunology department. So cytometry is kind of a workhorse method for immunology. And so we used and we developed in the beginning a lot of methods to analyze flow cytometry data, but now we're moving to other types of single cell omics data where we can even measure the full transcriptome at single cell level. And now the latest technologies that we have is spatial omics technologies, where you can even profile every cell at the single cell transcriptome level, but also find out the spatial location of all these transcripts. So that gives tremendous amounts of information. And of course, the challenge is then to develop novel models that can integrate these different types of data and of course get to novel insights. So what can we do with single cell data? Well, we can do a lot of interesting things, like trying to find out the structure that is present, for example, the different cell types, how these cell types are related to each other, try to visualize that in a comprehensive way. We can get more mechanistic insights into the system's dynamics and the underlying regulatory networks that are driving cell developmental processes. We can go one step further and also try to find out how cells are communicating with each other, both in a healthy state and in disease state. We can try to integrate all this data, for example, with clinical data, and try to do patient stratification. And of course, if you collect all these types of data for a large amount of patients, that really leads quickly to a very large amount of data, and you need big data technologies to still make it feasible to process all these amounts of data. So a lot of our research in the last 5 years has been in really automating the analysis of single cell data. Just going to show here a few examples on flow cytometry, where we actually developed a whole computational pipeline that consists of different steps, including automated quality control, normalization, automated cell type and identification, and then extracting properties from all these cell types that could be useful for predictive models, that could be used either for diagnosis, prognosis, or extraction of biomarkers. And then, of course, in the last step, you need to validate all these models in independent external cohorts. So first example that we worked on was the automated diagnosis of common variable immunodeficiency, which is one of the most frequently diagnosed primary antibody deficiencies. The problem with the diagnosis is that it's typically a diagnosis by exclusion. So typically it takes quite a while before the patient is diagnosed with rightly with CVID. And so we try to set out if we could develop models based on artificial intelligence to, from a blood sample, predict if a patient has CVID or not. So this was work done by Annelies Emmanuel together with the lab of Philomeen Haring at the UZ of Ghent. And so we collected a quite substantial data set here, and then we applied this pipeline for computational flow cytometry. And what you see in these tables here is that with the manual analysis, the classical way of analyzing flow cytometry, you would reach about 80% accuracy of the models. But using this automated flow approach, we could push that over to 90% accuracy in diagnosing CVID versus non-CVID, and also CVID versus other types of primary antibody deficiencies and healthy patients. Of course, these models are interesting because you can dive into them and see which features the model is actually using to make the prediction. And from that, you can extract useful biomarkers, like here some types of B cells or T cells that came out of these models as potentially interesting biomarkers. The second application is on myelodysplastic syndrome. So this was a collaboration with some people from the Amsterdam UMC. So there we also try to automate the process of diagnosing these patients with myelodysplastic syndromes. Many of these patients have a high risk of progressing later to acute myeloid leukemia. And so we set up a kind of similar pipeline here. The idea was also trying to automate the process, but also make it a little bit faster, because the typical manual workup in the hospital requires about 1 hour per patient to analyze all the flow data and do the diagnosis. So we also made a completely automated system here. We trained it on a training cohort. We validated on an independent validation cohort. And you see that also here we obtain very high numbers for the predictive performance of the models that also largely surpass the manual analysis. What was even more interesting is that using these 6 tubes of antibodies that were typically used in the clinic, we could actually reduce that to 1 single tube of antibodies that has almost the same classification performance, but needs a lot less antibodies. So it's a lot more cost effective and also needs thus less cells from the patients. So you have to take less bone marrow from the patients to have a classification model that has almost the same accuracy as the classical model. And very interestingly, also the time aspect of running the whole computational pipeline reduces the diagnostic process from 60 minutes, which would be the classical way of people doing it in the clinic, to 3 minutes or 30 seconds, depending on the computational method that we're using. So that's a huge saving in time. And for example, that would allow people in the clinic to maybe focus more their time on the more difficult cases. So also here we could extract some novel biomarkers from these computational models. I'm not going to go into detail, but some typical erythroid progenitor out, and they can be confirmed in the clinic as indeed being very important for the diagnosis here. So just to conclude my talk, I'm going to quickly give an overview of some of these single-cell RNA sequencing approaches. So in collaboration with the lab of Geert Bergs, we actually did a study on ovarian cancer, where we collected data from your ovarian tumor and also some sites of metastasis for a number of ovarian cancer patients. Was still a low number of patients here, 3 patients. And then we collected single-cell RNA sequencing before and after chemotherapy. And then the idea was to find out what is the heterogeneity of all the cancer and other cells that are present at these different sites of the cancer. And can we find, for example, typical marker genes for all these different populations? So you see here a UMAP plot, which is a 2D visualization of actually a very high dimensional data set, where you see the different clusters here representing the different cell types, so the cancer cells, the endothelial cells, and then different types of cancer-associated fibroblasts. And of course, all of them you can analyze more in detail because you have the full transcriptome profile. So at the right-hand side here, you can see which genes are specifically expressed then in these different subtypes. And of course, if you find these genes, you can actually use them later on as specific marker genes for example, specific populations that maybe can tell you something about chemo-resistant cells, and they could be used later on, for example, to design a novel flow cytometry panel so that you then can easily screen in a more cheap way these patients later in the clinic. I want to conclude by just a very short story about some novel methods that we have been developing on really trying to get more insights in the dynamics of intercellular communication, which actually nicely ties in with the network story by Professor Loscalzo. So we're developing here some novel computational techniques that allow us to actually reconstruct how cells are communicating with each other. So for example, if you have a cell 1 here that through a ligand receptor interaction is talking to cell 2 here, that then this receptor sets into action a whole regulatory cascade here, you can actually look at the transcriptional footprint in this cell 2 here and try to predict what was the ligand receptor interaction between cell 1 and cell 2 that was going on. So in order to do that, we combine, again, using a network representation, different types of information like ligand receptor interactions, signaling interactions, but also gene regulatory interactions. And we combine that in a very a large integrated network. And then we're using techniques like the Google PageRank algorithm to find actually influences of certain ligands to certain downstream target genes. And these downstream target genes can actually be seen as a footprint of maybe a potential ligand receptor interaction that is going on. So using this baseline network model here, we can actually combine that with a transcriptome of a single cell RNA sequencing experiment. And this would allow you to prioritize which ligand receptor interactions are actually active in the cell types that you're investigating. So this was published in a Nature Methods paper last year. And this is an example of this method in the liver. So in a nice collaboration with the lab of Martin Williams, we were looking at monocytes that are entering the liver and the niche signals that they get in the liver to develop into the macrophages of the liver, which are the Kupffer cells. And our long-term goal is actually to better understand how all these cells in the liver are working together to then later try to translate this into organoid models where we can actually develop in vitro liver mimics that, for example, would be very useful for all kinds of toxicity studies. So I would like to end by thanking all the people in my lab that contributed to all these different methods that we've been developing. And of course, also all the clinicians that we have been working with that were kindly enough to donate the data, but also to help in the interpretation of all the models. And I'd like to thank you for your attention and happy to take any questions.
Thank you, Ivan. Is there anyone? Yes, I see Professor Mack raising his hand. So, Professor Mack, the floor.
Thank you very much for the nice talk, Ivan. It was very interesting and very complementary to things that we do here. I had just a question. You started the presentation by speaking about different kinds of machine learning, and one of them is reinforcement learning. And my question is, in the different things that you are addressing today, do you already use reinforcement learning in your toolbox to solve problems for MDs?
No, we have not been using reinforcement learning so far. And also, if you look at the international literature, this is still kind of an underexplored area of machine learning because typically it requires some kind of interaction with
a reward function that is difficult to design.
Yes. Yes.
Okay, thank you.
People have been mainly looking at the other types of machine learning, so the supervised and unsupervised methods where you actually only need data, right?
Okay, thank you very much.
Zorba Ligon, you also have a question.
Thank you. So Yvan, you very nicely highlighted the logistic advantage and intellectual advantage of separating different cell types and characterizing the transcriptome on a single cell basis. And you use that to try and discern the different cell-cell communication pathways. My question is, obviously you could demonstrate some signatures of ligand receptor interactions that drive the expression of specific genes. But can you extend your model by going from the transcript to the actual protein and then to mediators that would be released in the intercellular milieu that would then feed back on other cells? And so in other words, do you think that the field is ripe now to go to single cell proteomics and try to move beyond the transcription regulatory network?
Yes, that's a very interesting question. Yes, all these other mediators that could be important for signaling between cells. So single cell proteomics is lagging a little bit behind with respect to what is currently possible for single cell transcriptomics. But people are working on that. So I've seen some first papers appearing last week, I think, from the group of Matthias Mann, who's very known in proteomics. So definitely people are working on that. And of course, that would be very interesting then to integrate with the transcriptomics data. There exists already some technologies to actually from the same cell measure transcript and protein information, but it's typically only surface protein like we typically do in immunology studies. But it will be certainly coming. Yeah.
Thank you.
And the last question will be for the president of the Academy, Professor Georges Casimir.
Thank you very much for your nice lecture. My question is there a need for special training today and a lack of biostatistician in the field of artificial intelligence and single cell data?
I think that that's a very relevant question, because now that all these methods are being increasingly used also by clinicians, well, one often heard critique that we are also faced with many times is that people in the clinic say, well, we don't trust these methods because we don't understand how all these methods are working. Mm-hmm. So of course, there is a need for training. We need to explain to these young scientists how all these methods are working. I think, maybe the role of the academy can be there to stimulate this more. But it's coming. I see all the young scientists, both from the clinical fields as well, they are also interested in learning how all these models are working. They're interested in analyzing their own data instead of sourcing it out to a bioinformatician. So I think things are slowly starting to change, but we can, of course, push that more and motivate them more.
Yes, it could be interesting to associate the universities to create new masters in this field.
Indeed, yes, thanks. And to also create novel research fields. I'm completely sure that will happen.
Thank you very much.
New projects for the academy and for you, Ivan. Thank you so much.